Hello everyone. Today I'll discuss about quality control and standardization of herbal drugs. So, quality control and standardization of herbal drugs ensure the safety, efficacy, and consistency of plant-based medicines by addressing issues like contamination, adulteration, and inherent viability in plant constituents. Key methods includes macroscopic and microscopic examinations, chromatographical separation technologies like thin lead chromatography, heavy metal analysis, and testing for volatile oils, ash content, moisture content, and so on. Standardization also involves implementing quality assurance protocols, chemical fingerprinting, and adherence to pharmacopoeial standards to guaranteed product quality and uniformity. So naturally, the question may arise to your mind, why do you need quality control study? Means what is the basic need for this? We need quality control study for several reasons, starting with the safety, which indicates the protects against contaminants like toxic metals, pesticides, and microbes. Efficacy, that ensures the presence and appropriate level of active compounds for consistent therapeutic effect. Purity and potency, which confirms the strength and absence of adulterants or other undesirable substances. Consistency, which refers to guarantees that different batches of the same herbal product are uniform. Then we are moving towards the next point, one of the major aspect of quality control studies or key aspects of quality control studies. It starts with physical evaluation, which is related with the macroscopic and microscopic examination that helps to identify the plant material and detecting foreign matter present within the system. Powder fineness, that ensures the material is properly ground for effective extraction. Moving towards chemical evaluation, where different chromatographical techniques which has been used, like different techniques including TLC or fingerprinting technique or any other technique, very much effective tool for separating, identifying those components present in a mixture. As value and extractive value, determining inorganic content and the amount of material extractable by solvents. In case of as value, we can determine easily the inorganic content present in it, and by extractive value, while we are performing extractive value, normally we are selecting three different solvents: ether, water, and alcohol. Water content or moisture content and volatile oil. Measuring moisture content and the quantity of aromatic or volatile components. Then moving towards contaminant testing, which includes heavy metals and pesticide residues. In heavy metals, we are using techniques to detect and quantify toxic metals like arsenic. In case of pesticide residues, we are analyzing for agricultural chemicals that may be present on the raw material. In microbiological contaminants, we are simply testing for the presence of bacteria and other microorganisms. What will be the standardization approaches for those? For this, we need to focus on some standard protocols, and that has been provided to some standard monograph as well as by different pharmacopoeias. So as we are talking about pharmacopoeial standards, we must say we are following guidelines from different official publications like pharmacopoeias which set quality standards for herbs and different kinds of herbal products in research as well as in different pharmaceutical industries or herbal industries. Chemical fingerprinting, which are helpful for creating detailed chemical profiles to identify and quantify key constituents, ensuring batch to batch consistency. Next, we must moving towards quality assurance, which is very much helpful to establish and follow rigorous testing and assurance protocols throughout the production process. Now, after performing or during performing such activities, some practicals, some maintaining the protocol, whenever we are doing all those, a question may arise to your mind that why we are doing this, what is the importance, what we will gain after doing this, means importance of quality control and standardization in herbal drug analysis. It talks about the same. So I'd say quality control and standardization are crucial in herbal drug analysis to ensure the safety, efficacy, and reproducibility of herbal medicines. Standardization involves establishing consistent methods for identifying, evaluating, and controlling the quality of herbal drugs and their preparations. This helps to minimize variations in active ingredients level, that helps to prevent contamination and ensure that each dose delivers the intended therapeutic effect. What is the key importance? First, ensuring safety and efficacy, which relates with the preventing adverse effects. Standardization helps to identify and eliminate toxic substances, adulterants, and contaminants that could cause adverse reactions or health complications to the patients. Whenever we are talking about consistent therapeutic effects, we must say it can be done by controlling the levels of active compounds. Standardization ensures that each dose of an herbal medicine has a predictable and reliable therapeutic effect. Avoiding misidentification. Proper identification of plant species is vital, as some plants may have similar appearances but different pharmacological properties or toxicity levels. Second, majorly, maintaining quality and reproducibility. It relates with consistent quality, reproducibility, quality control parameters. So when I talk about consistent quality, I must say standardization ensures that herbal products from different batches or resources have the same quality and composition, minimizing variations in potency. In case of reproducibility, standardized methods allow for the consistent production of herbal medicines, making it easier to replicate the desired therapeutic effects. And the moment we are mentioning about quality control parameters, we must say the various different kinds of parameters like botanical parameters, physical parameters, chemical parameters, biological parameters. Evaluations are used for standardization, ensuring a comprehensive assessment of quality. What about addressing challenges in herbal medicines? It's related to the complexity of herbal products, lack of standardization, need for analytical methods. In complexity of herbal products, we must talk about something which relates with the herbal medicines, and these are complex mixtures of various compounds, as we know, and their compositions can vary due to factors like plant species, growing conditions, and processing methods. In lack of standardization, I would say the lack of standardized methods and quality control in the past has led to inconsistencies in herbal medicine products, hindering their acceptance in mainstream healthcare. Whenever we talk about need for analytical methods, we must say modern analytical techniques like chromatography, spectroscopy, and microscopy are used to identify and quantify active compounds, aiding in standardization and quality control. Building trust and confidence, which talk about consumer confidence and regulatory compliance. So in customer confidence, it's related with the standardization builds trust among different customers or consumers, healthcare professionals, and regulatory bodies by demonstrating the quality and safety of the herbal medicines produced by different industries. Regulatory compliance says about standardization is essential for meeting regulatory requirements and ensuring that herbal products meet established quality standards. In essence, quality control and standardization are fundamental to the proper development, production, and use of herbal medicines. By implementing these practices, we can ensure that herbal remedies are safe, effective, consistently deliver the desired therapeutic benefits. Now, I will discuss about WHO guidelines, which is very much essential, especially when we talk about quality control and standardization of herbal drugs. In any of the independent nations, those are having their own constitution, those are having their own pharmacopoeia. These all independent countries, self-dependent countries, they basically set their rules and regulations for natural product industries or synthetic product industries according to their pharmacopoeia, and whatever regulations has been mentioning over there, it is something inspired by WHO guidelines or directly taken from WHO guidelines, because without WHO guidelines you cannot prepare a product, you cannot go for the testing protocols of a product, because WHO guidelines specifically meant for global safety, security and global acceptance of the product. It means if you are supposed to manufacture a product, a pharmaceutical product, whether it's synthetic or uh, herbal product, and if you're supposed to manufacture the product in India and if you want to market globally, so several parameters are there from where you have to collect the means permission. But one of the major aspect for this is whether you are maintaining WHO guidelines or not, because the moment you are thinking or going along with your product globally, moving towards create some global business along with your product, you need to focus on the guidelines, specifically WHO guidelines, because without following those, your product not even eligible to apply for the same for doing the global market. So I would say the WHO guidelines or World Health Organization guidelines present general consideration on potentially hazardous contaminants and residues in herbal medicines. It includes guiding principles of assessing quality of herbal medicines in terms of major contaminants and residues. It also recommends analytical methods for qualitative and quantitative determination of such contaminants and residues. The objectives of these guidelines are to provide quality control of crude drugs material, plant preparations and finished products, stability assessment and shelf life, safety assessment, documentation of safety based on experience or toxicological studies, assessment of efficacy by ethnomedical information and biological activity evaluations. The scope of these guidelines does not cover issues of adulteration of herbal medicines. It should be noted that these methods need to be validated for the material that is to be tested and also for each type of instruments. The other WHO documents and publications relating to the quality assurance of herbal medicines with regard to safety should include the following steps. The first one is authentication, which includes stage of collection, parts of the plant collected, regional status, botanical identity like phytomorphology, microscopical and histological analysis, taxonomical identity, etc. Foreign matters, where herbs collected should be free from soil, insect parts or animal excreta, etc. or any other foreign matter. Organoleptic evaluation, it includes the involvement of sensory characters the moment you are going to identify the product. Sensory characters include taste, appearance, odor, feel of the drug, etc. Tissues of diagnostic importance present in the drug powder. Ash values and extractive values, volatile oil, moisture content determination. Chromatographic and spectroscopic evaluation included TLC, HPTLC, HPLC methods will provide qualitative and semi-qualitative information about the main active constituents present in the crude drug as chemical markers in the TLC fingerprint evaluation of herbals. The quality of the drug can also be addressed on the basis of the chromatographic fingerprint. Determination of heavy metals. It includes the testing along with heavy metals like cadmium, lead, arsenic, etc. Pesticide residue. WHO and FAO, that is Food and Agricultural Organization, set limits of pesticides which are usually present in the herbs. These pesticides are mixed with the herbs during the time of cultivation. Mainly pesticides like DDT, BHC, toxaphene, aldrin cause serious side effects in human beings if the crude drugs are mixed with these agents. Microbial contamination, it includes usually medicinal plants containing bacteria and molds are coming from soil and atmosphere. Analysis of the limits of E. coli and molds clearly through light towards the harvesting and production practices. The substance known as alpha toxins will produce serious side effects if consumed along with the crude drugs. Alpha toxins should be completely removed or should not be present. In case of radioactive contaminations, microbial growth in herbals are usually avoided by irradiation. This process may sterilize the plant material, but radioactively hazard should be taken into account. The radioactivity of the plant sample should be checked accordingly to the guidelines of the International Atomic Energy, that is IAEA, in Vienna, and that of WHO. The quality of the raw material can be tested according to the following format. Name of the drug, it can be written by using the language, English language or regional name or exact botanical nomenclature. Part of the plant we were using, area of collection, distribution details, season of the crop, time and year of collection, pesticide and insecticides if it is needed during cultivation, condition of the drug, fresh or dry, form of the drug, powdered or intact, or cuttings like etc. In order to obtain quality oriented herbal products, care should be taken right from the proper identification of plants, season and area of collection, extraction, isolation and verification process. Chemical and instrumental analysis are routinely used for analyzing synthetic drugs to confirm its authenticity. In the case of herbal drugs, however, the scene is different, especially for polyherbal formulations, as there is no chemical or analytical methods available. The herbal formulations in general can be standardized schematically as to formulate the medicament using raw materials collected from different localities, and a comparative chemical efficacy of different batches of formulations are to be observed. The preparation with better clinical efficacy are to be selected after all the routine physical, chemical and pharmacological parameters are to be checked for all the batches to select the final finished product and to validate the whole manufacturing process. Thank you very much.